guys? Hello, everybody. Welcome. We are just about to get started here. Please bear with me as I work out some technical details. Sit back, enjoy yourselves. Got a fun one tonight. All right, guys. Thank you. Sorry, I did not mean to play that for another loop. So, I've been thinking a lot about, the purpose of these talks and the kinds of things that I want to kind of accomplish as we, as a crew of people, delve deeper into our understanding of science and technology and the cosmos, and where we fall within all of that. I know that there is an incredible problem these days, particularly with misinformation, a lack of clarity in our understanding of the sciences, and there seems to be a disconnect or an inability for us to commune with a polarized side of society that chooses to kind of cherry pick what they believe in or how they believe in things. So I've really wanted to try and spend the next couple of episodes exploring some interesting topics, how we can have more productive conversations around articulating facts that we can agree on collectively as a society, depending, no matter what side of the spectrum, the political spectrum that you lie on. And hopefully videos like this can help educate us a little bit. Not my videos, the videos that we're going to watch more specifically. So I think we're going to start off talking about the immune system. We're going to get a little bit into some cellular biology at some point. There'll be some miscellaneous tech articles that I want to go through. But hopefully in the next couple of episodes, we're going to have a epidemiologist and doctor of health policy on, who is going to help us interpret statistics and statistical studies, so that, as we move deeper into the conversations of doing your own research, and you see certain scientific articles or certain scientific journals getting posted, you as a layperson can maybe get some skills to be able to interpret not like the dense technical details of the study, but whether it's a good study or a bad study, or whether it's suspect, or whether it fits the criteria of something that is or is not good science. I think that is something that we as lay people can easily achieve, and hopefully through conversations like this, we're going to achieve that. So again, this is a conversation. I'd like to encourage people to chat, ask questions, chime in. We're going to talk about all kinds of different subjects today. And to kick things off, we are going to talk about the immune system. We're going to watch a great video from Kurzgesagt on how it works. It's a 3-part series. We're going to watch part 1 and 2. So why don't we start with diving into it?
The human immune system is the most complex biological system we know after the human brain, and yet most of us never learn how it works, or what it is. Your immune system consists of hundreds of tiny and 2 large organs. It has its own transport network spread throughout your body. Every day, it makes hundreds of billions of fresh cells organized like an army, with soldiers, captains, intelligence officers, heavy weapons, and crazy suicide bombers. It's not some sort of abstract entity. Your immune system is you. Your biology protecting you from the billions of microorganisms that want to consume you, and from your own perverted cells that turn into cancer. It's so manifold that it's impossible to cover in one video, so we'll make a series looking at different aspects of it. Today, what happens when your body is invaded and your first lines of defenses are engaged in a fight for life and death?
So as we progress into the video, we're going to talk a little bit about hopefully maybe taking some opportunities to delve into more specific pieces of the science. But what's really important is this is going to talk about the immune system as it exists today. And hopefully that is going to help us bridge a conversation into how vaccines work inside said immune system, and starting to understand like traditional vaccines that we can't really question whether or not they're safe or effective. Things like the polio vaccine, measles, mumps, and rubella, etc., etc. So.
It's been a normal day when suddenly the world explodes and an asteroid rips the sky open. Countless alien life forms invade, ready to destroy cities and infrastructure and eat civilians. Or this is what your cells experience. You look at your bleeding thumb that you just cut on a dirty twig in the park. How annoying. But inside the wound, a horrible catastrophe has happened. There are dead cells and blood and dirt everywhere. Even worse, countless bacteria invade the warm caverns between your helpless cells to explore their new home, steal your resources, and poop everywhere.
So this is the effect, obviously, when you cut your hand, it gets red and swollen. That's your immune system responding to the bacteria that have invaded. Because if nothing got in, it would just be a clean cut and heal really quickly. But it gets irritated and gets pussy and gross. That's because the bacteria that have gotten in are starting to kind of win the war.
Immediately, the first stage of your defense kicks in. The cells that survived the impact or are hurt or dying scream in panic, releasing an onslaught of chemical alarm signals that awaken your immune system. The first cells to show up are macrophages. If an average cell were the size of a human, a macrophage would be the size of a black rhino.
Wow.
A stoic cell in principle, but you wouldn't want to annoy it. Bacteria do annoy them. Within seconds, the large cells attack and begin killing them without mercy. They stretch out parts like the arms of an octopus and grab the bacteria to swallow them whole and digest them alive.
So one thing I just really want to quickly touch on is that the majority of every piece of this song and dance is being performed by proteins or collections of proteins. We touched on what protein folding is in the AlphaFold episode. We're going to talk about it, I think, later in this tonight's talk. But it's really important to understand that the little pieces of working machinery, the bits that are doing the tearing and facilitating these functions inside of cells, are actually those specialized proteins that are synthesized from your DNA.
A macrophage can eat 100 bacteria before it's exhausted. But there are too many enemies, so the macrophages call for reinforcements. In your blood, hundreds of thousands of neutrophils pick up their signals and move to the battlefield. Neutrophils are intense suicide warriors that only live to kill. They're so enthusiastic about killing that they kill themselves a few days after birth, so they don't have time to accidentally destroy your body from the inside. As soon as neutrophils arrive, they begin vomiting deadly chemicals at bacteria or devour them. They are so careless in their attacks that they are causing real damage to your own cells.
So keep in mind, like what's happening here is there's a there's a first line of defense, which are the macrophages, and if they can get the bacteria infection under control, then no harm, no foul. Everything's fixed. However, if they release those signaling proteins out into the body, which brings in the next level of defense, this is a level of defense that is significantly more damaging to cells. The macrophages just grab bacteria and eat it and chew it up. These, like they said, are kind of like suicide bombers. They're just blowing up and obviously doing kind of how chemotherapy works in a certain degree. Like, it's attacking the cancer, but it's also attacking your body. So these neutrophils though, are doing, a lot of damage against the enemy, there is, what do you call it, more collateral damage being, as a result.
But collateral damage is not their concern now. Or
Sorry.
ever. Some neutrophils go so far to push their suicide button and explode, casting wide and toxic nets made from their own DNA, filled with deadly chemicals that trap and kill bacteria. Sometimes they can continue fighting after that, even though they're sort of dead already. This is how much fun they have killing. While the battle rages, your blood vessels let fluid stream into the battlefield like a dam opening up towards a valley. You notice this as inflammation. Your thumb swells up a little and gets red and warm. The fluid brings a silent killer into the battle zone, millions of complement proteins, a sort of automated liquid weapon that stuns and kills bacteria by ripping holes into them. We made a whole video explaining them in detail. We're reaching a crossroad now. If things go well, your first line of defense kills the invaders quickly. But sometimes the enemies are too strong and would overwhelm your defenses eventually, which means certain death for you, the human. This is the hour of the dendritic cell, your immune system's intelligence officer. While your soldiers were bashing in heads, it was collecting samples by ripping bacteria into tiny parts and covering itself in it. Like a soldier decorating itself in the guts of a dead enemy. The cell leaves the battlefield and enters the superhighway of your immune system that connects all your tissues with your immune headquarters, your lymph nodes. The dendritic cell coming from the battlefield is looking for a helper T cell, which is a sort of all-purpose commander cell within your immune army. But not any helper T cell, one that happens to have just the right weapon for the bacteria that infected your wound.
So this is a fascinating part about the immune system. So again, like all of the biomechanical machines, if you kind of reduce them down, their constituent elements are just proteins, which are, little things that have a particular shape, and that particular shape does a particular thing. And so all of these T cells that are in your body have different, permutations, different tools. So like, the proverbial sword or an axe or a bow and arrow. And those mechanisms, those action potentials work better against some enemies or some type of diseases than others. So the idea is that your body is able to amass these memory cells which contain instructions for how to mass produce the particular type of weapon that is required to respond to that particular threat.
So it goes around and rubs itself, still covered in bacteria parts, against every helper T cell it meets. Most T cells are a bit disgusted and not interested, but after a few hours, something clicks. A helper T cell recognizes the bacteria parts. This cell is the weapon that's needed right now. The dendritic cell is overjoyed and activates the helper T cell. Okay, wait. How come your immune system has a cell that has a weapon against the specific bacteria that infected you? Well, your immune system has a perfect weapon against every possible disease in the universe, against the Black Death, the coronavirus, or an infection that will emerge in 100 years on Mars. We'll talk about this a bit more in the next video because it's very
That's coming
complex.
up after this.
So for now, just know that you have billions of unique helper T cells that each have weapons against every possible enemy. After the right T cell is activated, your second line of defense awakes and rises like a teenager that needs to get up on a school day, very slowly. Your heavy weapons are incredibly effective, but they're not fast. The activated helper T cell begins to clone itself over and over again. 1 becomes 2, 2 become 4, until there are thousands of them. Now they split into 2 groups. The first group quickly moves to help out your soldiers. At the battlefield, things are getting out of hand. A tired macrophage is ready to give up. After fighting for days, it just wants to go to sleep, like many of its buddies have done already. But now the helper T cells arrive. One of them comes to the tired macrophage and whispers something using special chemical signals. In a heartbeat, the demoralized soldier feels fresh again. But there's something else, a hot, white anger.
So remember, all of this communication is done through the process of releasing different kinds of proteins, signaling proteins, which the cells have like receptors for. So typically, a lot of these interactions are always described as a lock and a key, and that's very much what it's like. These things just bump and rub against each other unless some of them lock, and the locking mechanism is very specific, right? And so cells that are meant to bind together will find each other, they will lock together, and then they will stimulate each other in a particular response. So that's how these signals are being pushed in. This is a really important point that I want to stick with people, because the production, if we have the ability to synthesize, like to be able to discover and synthesize these proteins, it's a game changer for medicine, and we're going to have a lot to say about that in a little bit.
Macrophage knows what it needs to do. Kill. Invigorated, it throws itself against the enemies once again. All over the battlefield, this begins to happen. Meanwhile, the second group of helper T cells was working on activating another line of defense, B cells, your antibody factories. Antibodies are protein superweapons that look like tiny crabs with two pincers to grab enemies. Just like the helper T cells, there are B cells in your body that are able to make just the right antibodies for every possible enemy. And the helper T cell is looking for exactly these B cells. After a day or two, the right B cell is found and begins to clone itself. As soon as enough clones have been made, each B cell begins pumping out up to 2,000 antibodies per second. About a week after you injured yourself and bacteria invaded, your second line of defense finally arrives in full force.
Takes a while.
The tiny army begins to saturate the battlefield, pinching and stunning desperate bacteria. The antibodies clump them together and make them unable to move or fight, while your soldiers massacre the defenseless victims. The tide is turning fast.
This is sick.
As the last enemies are cleaned up, your soldiers realize they are no longer needed and begin to kill themselves to save resources. But not all of them. A few helper T cells remain and turn into memory cells. They will guard the tissue for years, making sure the same bacteria will never again gain a foothold here. Similarly, a few B cells will stay alive and keep producing a low amount of antibodies, making you immune against this bacteria, maybe for the rest of your life. One day you wake up and notice that the wound has grown over and left nothing but a faint red mark. You were completely unaware of the drama your cells had to deal with. For you, the whole ordeal was a slight annoyance, while for millions of cells, it was a desperate fight for life and death. But this is just the beginning of the epic story that unfolds inside you every day and is told in full in Immune.
I highly recommend people get this book if you're at all interested in this subject. It looks incredible. Definitely gonna get myself a copy. Very cool stuff. We are gonna look at part 2, which I actually haven't seen yet, so I'm very excited to watch with you guys. But one of the things that we are also going to look at is Lex. This guy.
Powering.
Yep, sorry. This episode with Vincent Racchianello is incredible. I'm very excited to get to this. I think a lot of what we're about to watch is going to inform our understanding of some of these processes. There's a couple subsequent supporting videos we can watch as well. But why don't we dive into part 2 of the Immune video, and then we'll start really getting into the hot stuff.
Every living being needs to fight off other living beings that want to feast on them. So as multicellular life evolved over billions of years, it came up with ways to defend itself. Today, humans have a sophisticated defense network, like physical barriers, defense cells, and weapons factories. But one of the most important defenses of our body is largely unknown: the complement system. It evolved over 700 million years ago and is an army of over 30 different proteins that work together in a complex and elegant dance to stop intruders. All in all, about 15 quintillion of them are saturating every fluid in your body right now.
It's a lot.
Guided by nothing but chemistry, these proteins are one of the most effective weapons we have against invaders. Many other parts of the immune system are just tools to activate the complement system. But it's also really dangerous. Imagine having trillions of little bombs inside your blood that could go off at any moment.
Wow.
So our cells use numerous mechanisms to prevent complement from accidentally attacking them. Okay, what exactly does it do, and what makes it so dangerous?
This is going to be an interesting one for sure. I'm excited. And guys, remember, if anyone has questions or wants me to
In a
elaborate
nutshell,
on things, just
the
let me know.
complement system does three things. It cripples enemies, it activates the immune system, and it rips holes in things until they die. But how? After all, these are mindless proteins randomly drifting around without will or direction. Well, this is actually part of the strategy. Complement proteins float around in a sort of passive mode. They do nothing until they get activated and change their shape. In the world of proteins, your shape determines what you can and cannot do, because shape determines what you can interact with and in what way.
You're just talking about
For
this,
example, in your passive shape, you might do nothing. In your active shape, however, you might, for example, change the shape of other proteins, activating them so they can activate others. Mechanisms like this one can start cascades that spread very quickly. Imagine the complement proteins as being like millions of matches very close together. Once one catches fire, it ignites the ones around it. They ignite more, and suddenly you have a big fire. To show the actual mechanisms of the complement system is a tad complicated and overwhelming, so we'll simplify here.
Okay.
Now, let's imagine you cut yourself and a bunch of bacteria enter the wound and make it into the surrounding tissue. Our complement attack begins with C3. C3 is the first match, the initial spark that will start our fire. And to do that, C3 needs to switch from passive to active. How this happens is complex, but let's just say it can happen randomly through other complement proteins that bind to enemies or through antibodies. All you really need to know is that C3 breaks into two smaller proteins, C3a and C3b, that are now activated. One of these parts, the C3b protein, is like a seeker missile specialized in bacteria, fungi, and viruses. It has a fraction of a second to find a victim, or it will be neutralized by water molecules. If C3b does find a target, it anchors itself very tightly to its surface and doesn't let go. By doing so, the protein changes its shape again. In its new shape, it's now able to grab other proteins and start a small cascade, changing its shape multiple times, adding other complement proteins to itself. Finally, it transforms itself into a recruiting platform known as C3 Convertis. This platform is an expert at activating more C3 proteins that start the whole cycle anew. An amplification loop begins. Soon,
It's almost like it colonizes the surface of the bacteria. So once one of those proteins gets activated, like the match example, it starts a chain reaction that's able to have more C3 cling to the surface, activate, and continue the cascade, right?
thousands of proteins cover the bacteria. For the bacteria, this is very bad. It can cripple the bacteria and make them helpless or slow them down. Imagine being covered by thousands of flies. But there's more. Do you remember the other part of C3? The C3a protein. C3a is like a distress beacon. Thousands of them flood away from the site of battle, screaming for attention. Passive immune cells notice the C3a proteins and awaken from their slumber to follow the protein tracks to the site of infection. The more alarm proteins they encounter, the more aggressive they get. This way, complement guides reinforcements exactly to the place where they're needed the most. So far, the complement has slowed down the invaders and called for help. Now, it's beginning to actively help to kill the enemy. The first immune cells to arrive at the battlefield are phagocytes, which means cells that swallow you whole, trap you in a tiny prison, and then kill you with acid. But to swallow an enemy, they need to grab it first, which is not easy because bacteria prefer not to be grabbed and are sort of slippery. But now the complement that has anchored itself to the bacteria acts as a sort of glue that makes it easy for the immune cells to catch their victims. But it gets even better. Imagine being...
So it's like literally embedding hooks that are making the phagocyte or like the macrophage or whatever able to grab onto it much easier. So imagine, you know, like you're trying to pick up like a slippery ball of ice, and then all of a sudden you have like a glove that has like sharp spikes on it. It's gonna be pretty easy to pick up, right?
Being covered in...
Or I think Velcro. Velcro's a much better example, right? It coats one end with the little hooks, and your end does the other hooks, and they're just super easy to click together.
Again. Now, imagine them turning into wasps. Another cascade is about to begin. On the surface of the bacteria, the C3 recruitment platform changes its shape again and begins to recruit new proteins.
C5,
Together,
C5b.
they begin the construction of a bigger structure, a membrane attack complex.
Okay.
Piece by piece, new proteins, shaped like long spears, anchor themselves deep into the bacteria's membranes, until they rip a hole into them that can't be closed again. Fluids rush into the bacteria and their insides spill out. They bleed to death. The remaining bacteria are maimed and distracted by the complement and quickly taken care of by the arriving immune cells. The invasion has been nipped in the bud before it had the opportunity to become dangerous. You probably didn't even notice it. But while bacteria are not happy about complement, the enemies it might be the most useful against are actually viruses. Viruses have one problem. They need to travel from cell to cell. Outside of cells, they're basically hoping to randomly bump against a cell to infect by pure chance. Here, they're completely defenseless. And here, complement is able to intercept and cripple them so they become harmless and guide the immune system to devour them.
Nice.
Without complement, virus infections would be a lot more deadly.
Interesting.
But wait, if we have such an effective weapon, why do we ever get sick? The problem is that in a war, both sides adapt. For example, when the vaccinia virus infects a cell, it forces it to produce a protein that shuts complement activation down.
Interesting.
This way, the virus creates safe zones around the cells it infects. When it kills them and tries to infect more, it has a higher chance of being successful.
Okay.
Or some bacteria, for example, can grab certain molecules from the blood that keep the complement system calm and make themselves invisible.
So these are adaptations, right, that we're seeing in viruses and bacteria as they infect people, right, and are able to survive. Typically, adaptations happen through mutations. There's like a random, like, weird like aspect of that particular organism that gives it a particular advantage, and then as it divides, that advantage is passed on, you know, down its lineage. And this is how things like viruses are able to mutate. And this is why, you know, at some point I think it's going to be important as we're talking about, you know, vaccines and why it is so important to immunize populations against these pathogens, is the more they are allowed to rip through people, the more mutations. That's why we have, you know, with COVID, we have the Delta variant right now, that literally could have been avoided. Like, I mean, it's tough to, but, you know, had we been able to stop, quote unquote, the spread and actually, you know, reduce the transmission, there might have been some people that weren't infected where it didn't have the opportunity to mutate. We wouldn't have more like virulent strands of it. So this is why it's important to understand like the process of mutation, why mutation gives like an adaptational advantage. And why it's important that when we're talking about warfare, you know, we're talking about strategy and against a particular pathogen, you know, we need to understand how it arms itself against us and how it's responding to, you know, our attacks. Mutation is one of them. It's why it's really important to get it under control.
So the complement system, while being extremely important, is only one player in the complex and beautiful organization that is our immune system. A beautiful example of how many mindless things can do smart things together. This video was sponsored by you. Without your direct support, this channel would not exist, and it's hard to overstate how grateful we are for that. In 2019, an average Kurzgesagt video took at least 1,200 hours to produce.
That's wild.
We need months to research and write our scripts, clear them with experts, illustrate and animate every scene from scratch, and to compose original music. This is only possible through a combination of sponsorships, your support on Patreon, and selling merch, like this poster and postcards we made about the compliment system. If you yourself want to support us, you can do so by watching, becoming a Patreon, and maybe getting a personal bird, or by getting one of our calendars or posters. The purpose of Kurzgesagt is to make the best videos we can, and to make science more approachable. Thank you so much for making this possible.
Okay, guys. Sick video. Where is it here? There is a video that I'm looking for on Khan Academy. Just give me 1 sec. I wish I had it queued up. Okay. We are going to dive into a very interesting conversation between Lex Friedman and this gentleman, and they are going to talk a little bit about vaccine efficacy, how we think maybe, bacteria sort of evolved, a wide variety of topics. The podcast is almost 3.5 hours. I don't think we're going to have time to get through the whole thing tonight, but we'll definitely, get started. We're going to jump into a really interesting conversation around how bacteria might have entered our cells and became like a symbiotic element of our cellular biology and a lot of organisms' cellular biology. So let's jump in.
What would you say is the magic moment along that line? I saw it was 1 or 2 billion, maybe 3 billion years it took to go from bacteria to like complex organism. Like, it seems like Earth had a very long time, like not a very long time without life, and then a very long time with very primitive life. Maybe I'm discriminating calling bacteria primitive life.
Yeah,
but
people would object to you
yeah.
doing that for sure.
But it seems like complex organisms, when you starts becoming something like, I don't know what's a good, not animal-like, but more complexity than just like a single cell. What do you think is the magic there? What's the hardest thing if you were trying to engineer Earth and build life and build the simulations? Obviously, we're living in a video game, what this is. So if you're trying to build this video, what's the hardest part along this evolution pathway?
So bacteria are mostly single cells. They do make colonies. They get together in biofilms, which are really important. But they're all single bacteria in that. And the key is making an organism where cells do different things. We have skin cells and eye cells and brain cells. Bacteria never do that. And the reason is probably energy. Bacteria can't make enough energy to do that. And so there was another cell existing at the time, the archaea. And the idea is that a bacteria went into an archaea and became the modern day mitochondria, the energy factory of the cell. And that now let that cell develop into more and more complicated organisms.
So the mitochondria, like he said, is the powerhouse of the cell. It is where your cell, if I remember correctly, it converts energy, glucose into energy, and allows it to produce the different things the cell needs to live. I know I'm being super technical here. The theory is that the mitochondria was a bacteria that was particularly powerful and particularly good at producing energy, and it essentially achieved symbiosis with the cell, which was the predicate for the ability for complex life to evolve. Complex life, like life typically was very simple because it was all about, the conservation of energy. But when you have a much stronger capacity to produce more energy from like something like a mitochondria, all of a sudden your ability to specialize and diversify, different types of cells for different types of organs, that's where sort of the complexity of life has come from.
Like we have today, it was all about energy.
So the mitochondria, the energy, the mitochondria is the magic thing.
I think so. It's just actually not my idea. It's Nick Jones. Have you heard of Nick Jones? He's an evolutionary biologist in the UK, and he's done experimental work on this. And it's his idea that the defining point was the ability to make a lot of energy, which a mitochondria can do. It's basically a whole bacteria inside of a bigger cell, and that becomes what we now call eukaryotes, and that they can get more and more complicated. So let me bring you back to the viruses. I want to finish that story.
Yeah, at which point do viruses come along?
So remember, we have these precellular, they're called precellular replicons, right? And so we have a precellular stage where we have these self-replicating molecules. Then cells arise, and then the self-replicating molecules invade the cells. Why? Because it's a hospitable environment. I mean, they didn't know that. They just went in, and it turned out it was beneficial for them, so it stuck. And they replicate inside the cell now, where they have pools of everything they need. They get more and more complicated, and then they steal proteins from the cell to build a protective shell. Right. And then they can be released as virus particles. They're now protected. They can move from host to host, and because they're at the earliest stages of cellular evolution, they can diversify to infect anything that arises. And that is why I think there's so many of them, and everything on the planet is infected. Because the ancestor of everything was infected many years ago.
So it's easier to steal than to build from scratch.
Totally.
So like it's easier to sort of break into somebody else's thing and steal their proteins.
Yes. My colleague Dixon Despommier calls viruses safe crackers.
Safe crackers.
Well, I mean, like, you know, when a tiger eats like an antelope, right? Or a lion eats an antelope, like, that's literally what they're doing. They're stealing the energy from another organism. They're like, hey, you built all that muscle and all that protein and all those minerals that are all in your stuff. I'm taking that, right?
Crackers. So
it's
it's
a
just
predicate for life.
From an evolutionary perspective, it's easier to steal because you can select, but then you have to figure out mechanisms for stealing, for breaking into, for cracking the safes.
Well, you don't have to figure out. It just happens, right? Because molecules are so diverse that a molecule gets into a cell, and if there's a protein that sticks to it, it's going to stick, and that gives an advantage. There's no,
this is what we were just talking about before, right? These are these random occurrences, mutations, or just proteins happening to bump into each other, binding in a certain way, giving an advantage, and it being able to do a certain functionality. Like that is again like the predicate for a lot of the way evolution works.
There's no planning. There's no thinking about it, right? It just happens.
Oh, we'll return to that. But these numbers are crazy. So what, as these more complex organisms evolved, let's take us humans as an example. Should we be afraid of these high numbers? Should we be worried that there's so many viruses in the world?
But to a certain extent, I mean, it's twofold. They're good and bad, right? Viruses there's no question they can be bad. We know that because they've infected and caused disease throughout history. But you and I are full of viruses that don't hurt us at all.
This is another excellent point, is that there is an enormous number of viruses that actually just are incompatible with getting into our bloodstream or affecting human beings. He brings up a really good example shortly about how if you check people's like poop, there is an enormous amount of X virus and B virus because like that's what's in like peppers and lettuce, and it's just not something that like goes into the human system. They'll elaborate
and
here.
every organism is the same, so they are clearly beneficial as a consequence. So I think every living thing on the planet has multiple viruses infecting everything you can see, and most of them I think we don't worry about because they can't infect us. They're unable. In fact, now you can actually take your feces and send them to a company, and they will sequence your viruses in your feces for you, your fecal virome, right? And the most common virus in human feces is a plant virus that infects peppers. It's called pepper mottle mosaic virus. And that's because people eat a lot of peppers. Mm-hmm. And it just passes right through you. Cabbage is full of viruses from the insects that walk on the cabbage in the fields. We eat them. They just pass us. So I think most of the viruses we don't need to worry about, except when we're talking about species that are closest to us, mammals, of course.
Mhm.
And I think the most numerous ones are the most concerning. Their viruses, like bats. Bats are 20% of mammals, and rodents are 40% of mammals. And we
Wow.
humans live nearby, right?
Yeah.
And we know throughout history many viruses have come from bats and from rodents to people. No question
So
about
there's
it.
a prox.
So we're I don't think we're going to spend any time getting into the whole like the where this latest virus has come from, but there is legitimate like evidence that people have transmit viruses from bats and rodents. Like look at the Black Plague, look at things like rabies, and the reason why that is possible is because of the genetic similarities, the biological similarities between like rodents and bats. That's why why do you think we test things on mice? Right, we test drugs and different chemicals, and when we tested and he talks about this, like when we test like the vaccine that uses like the nanolipid particles on mice, and you like overdose them on it, like yeah, it's not good for them, like it fucks them up, but like that's thousands of times what you would put in a human or in blah 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 blah. So anyway, ranting here.
Proximity in terms of just living together and a proximity genetically too, so it's more likely that a virus will jump from a bat and a rodent.
And birds too. Birds can give us their viruses. That's happened. Influenza viruses come from
Bird
birds
flu.
mainly. So I think those are the three species. Not species. It's higher than species, obviously.
Yeah,
but
the
those are the three I
groupings,
would worry
yeah.
about in terms of getting their viruses. And we don't really know what's out there, right? We have very little clue about what viruses. And I've for years wanted to capture wild mice in my backyard and see what viruses they have, because no one knows.
We,
And it's an
we
easy.
can't ask them. So you mean map? Like, is there
I
a way
can't
to
ask them. Yeah. No, would have to sacrifice them and take tissue and then bring it in the lab and do genome
So
sequencing.
you can do a thorough sequencing to determine which viruses. Is there a sufficiently good categorization of viruses where you'd be?
That's a very good question. So whenever you do sequence, right, you get some environmental sample and you extract nucleic acid and you sequence it. What you do is you run it past the database. Right, the gold standard is the GenBank database, which is maintained here in the U.S. And you see if you get any hits. And then you can say, look, this sequence is similar to this virus, and you can classify all the viruses you see. The problem is 90% of your sequence is dark matter. It doesn't hit with anything. It's probably a lot of it is unknown viruses, and that's going to be hard to figure out because someone's going to have to go after it and sort it through. So yes, you can find a lot of viruses, and the numbers you get are astounding. You can find thousands of new viruses just by looking in various life forms, but there are many more that we don't pick up because they're not in the database.
Maybe this is a good time to take a quick tangent. What do you think about AlphaFold 2? I don't know if you've been paying attention to that.
So we talked about AlphaFold a couple of times. We're going to keep talking about it, hopefully, because there's going to be some like absolutely incredible things that come out of it. But AlphaFold obviously solved the protein folding problem, and essentially the potential output of this breakthrough is that we will be able to create and understand how proteins are folded with higher degrees of accuracy, leading us to being able to correct an enormous number of medical conditions, produce completely revolutionary treatments for things like cancers. And with the delivery mechanism of mRNA, like they're already testing things, they're already testing a vaccine against HIV. There are certain cancer vaccines or cancer treatments that will, create a strand of mRNA that creates a particular type of protein that will stick to tumors and have your immune system just attack them and shrink them and destroy them. So we'll get more into sort of that process and how that stuff's going to work a little bit later, because these guys elaborate on it really well. But here's more on AlphaFold 2.
DeepMind solving the protein folding problem, and then also releasing, first of all, open sourcing the code, which is, for me as a software person, I love. And then second of all, also making like 300,000 predictions or something like that for different protein structures and releasing that data.
Yeah.
So on the side of, because you're saying there's dark matter,
Right.
like, is there something what are your general thoughts, level of excitement about their work, and second, how can that be applied to viruses? Do you think we'll be able to explore the dark matter of virology using
Yeah,
machine learning?
absolutely. Because in all this dark sequence, you can translate it and make a protein. You can see what a protein looks like. It has what we call an open reading frame, right? A start and a stop. And right now, it's just a bunch of amino acids. But if we could fold it, maybe the fold would be like something we already know, some protein fold, which gives you a lot of clues, right? Because there are only so many protein folds in biology, and that dark matter is probably one of them. So I think that's very exciting because for years I followed structural biologists for years, and you know, in the beginning we couldn't even solve structures of viruses; they're too big. We could do small molecules like myoglobin, like that was the first one done. It took years to do that. And then, as computational power increased, then they could start to do viruses, but it took a long time. X-ray
Interesting.
crystallography depended on getting crystals of the virus, right? And
we
now,
we talked about X-ray crystallography and how expensive it is. It's an incredibly laborious process. It takes like sometimes up to a year to actually fully go through the whole process. And it essentially it involves isolating, the protein in a solution, and then using, a whole bunch of magic and chemistry to crystallize that solution. And what happens is those proteins get captured in these crystals, and they can use X-rays to pass through the crystals and then map out the three-dimensional structure of those proteins or of those viruses. So X-ray crystallography is an incredibly powerful tool, but it takes a long time for it to actually do its thing, and it's very expensive. So the concept of being able to take a computer and point AlphaFold at something like that and figure those things out for us would be absolutely huge.
we can do cryo-electron microscopy, which is much faster. You could solve the spike of SARS-CoV-2 was solved in 2 months by Jason McClelland here in Austin, actually, at the beginning of the pandemic. But you're limited. You can't do huge proteins. You can only do moderately sized ones. So, actually, you can do viruses, but you can't do small proteins. So that speeded it up, but it's still too fast to solve. You get a new protein, you want to solve its structure. So if we could predict it, and I know from talking to structural biologists, this has been their holy grail from day one. They want to be able to take a sequence of a protein, put it in a computer, and have the structure put out without having to do all the experiment. So that's why this is very exciting that you can predict it. I mean, it's not finished, obviously, and there's more to do. But I think there will be a day where you could take any amino acid sequence and predict what it's going to look like.
See, but like, aren't structural biologists going to get greedy? So once you have that, don't you want to go more complicated then? Don't you want to go, because that's just the first step, right?
Sure,
to go from amino acid to the structure.
Yeah.
Then there's like multiple protein interactions. Like, how do you get to the virus?
Well, so that's what the ultimate goal of getting a structure is, that then you can do experiments and figure out what the structure means, right? So in the old days, structural biology was a career in itself. You worked with people who had a system and you solved proteins for them, and then you moved on to another one. You didn't really do any experiments. The other people got to do all the interesting experiments.
Right.
So it's like one group of people was discovering and one people was doing the experimentation. We're going to skip ahead for a second because the next part I think is particularly interesting for folks to know about, and we're going to talk a little bit about SARS-CoV-2, influenza, vaccines, how does mRNA work, and is it safe? Hopefully we can cover all of this tonight. We'll see how it goes. But we're going to talk about now the most terrifying virus, which I think is a phenomenal conversation and introduction to rabies and how the rabies virus works, how the vaccine can be given to you after you have contracted it, why that is. Let's get into it, guys.
Right.
What would you say is the most fascinating, terrifying, surprising, beautiful virus to you?
That's such
of
an
all the viruses you look
I
at,
just gotta say, that's so fucking Lex. That's such a Lex thing. What is the most horrifically terrifying yet elegantly beautiful thing you've ever seen? I really hope he asks me a question like that when I end up on his podcast someday. Wishful
sometimes
thinking
when you
alert.
just sit late at night with a glass of wine, looking over the sunset, which virus do you think about?
So fulfilling all of those adjectives is hard, right? Fascinating, exciting, terrifying.
Well, the terrifying is an optional one, I think, because maybe that puts a lot of pressure.
I see. Terrifying is, to me, I'm not terrified because I think we can handle as most viruses, as you see with this brand new one that emerged a year ago. We can handle it.
From a virology perspective.
Yeah, I mean the human perspective is a different story, right? That's always an issue. But so I think there are a couple of different categories of virus. So we could do the terrifying, and I think rabies is a terrifying virus because unless you're vaccinated, 100% certainty you're going to die. So you get bitten by a rabid raccoon or bat or dog, whatever, and and there's still 70,000 deaths a year of rabies throughout the world because there are a lot of feral dogs running around that are infected. Unless you're vaccinated, you're going to die. There's nothing we can do. But we do have a vaccine which we can actually give you even after you've been bitten, which is the only vaccine that works that way. It's a therapeutic, right? It will treat your illness because the disease takes so long to develop. Eventually you get all kinds of neurological issues and paralysis and so forth, but it takes time, and you can be vaccinated and it will prevent that in the meanwhile. So
That's like fucking crazy. Like that's one of the most amazing things about rabies. It's horrific, and like I would never subject the viewers to it, but if you're interested and you look up what the effects of rabies are on people and you see someone suffering from it, it's a horrifying way to die. And if you ever get bit by something, like, go get that shot, because it's a miracle of modern science that we're able to get vaccinated while it's incubating in our system.
people always say, what's the most lethal virus? Is it Ebola? I said, no, it's actually rabies. Unless you're vaccinated, it will kill you.
But maybe it's good to linger, because we'll talk about
100%
vaccines
fatality rate.
a few times today.
100%.
It's good to linger on cases where vaccines have clearly, undoubtedly helped human civilization.
Mm-hmm.
And it seems like rabies is a good example.
Oh, rabies is great because everyone knows what happens when somebody gets rabies, right? You have fear of water, hydrophobia, your body becomes spastic and stiff and jerks around, and you lose consciousness. You can't, no more.
It's not a fun ride to death.
It's horrible. It's a horrible way to
fucked up,
die.
man. It's awful.
So I think most people know that. It's been popularized enough in media, right? So that nobody would probably object to getting, I was just bit by this raccoon and it ran off. Okay, well, we should assume it's rabid. We should immunize you. And most people are okay with that.
Yeah.
Right.
Because they know the consequences. And it's also pretty rare, right? It's not like something that you're trying to get into the arms of, 250, 300 million Americans. That's hard. But the few thousand every year, it's easy.
So the transmissibility is difficult, right? It has to. Oh, it's not airborne. So
It's not airborne. It's just you have to be bitten. Although some people claim you could walk into a cave, and the bats, breathing out rabies virus could infect you. But I don't really think that's well substantiated.
Yeah.
I think it's a bite.
How would you do a study on that?
Yeah, it's very hard to do. You'd have to collect the vapors in the cave and show that they're infectious, and by the way, someone emailed me the other day, you'll like this. They say, why can't we just immunize all the bats in the world against these viruses? And I said, well, how would you do that? There are caves everywhere, right?
Yeah.
He said, well, maybe you could just go and aerosolize that.
Yeah.
It's pretty dangerous.
And then all the bats should have vaccine passports to make sure that they're
Yeah,
all...
I said you have to get their consent before you do it. But we do.
This is what he is about to say, I think, is an incredible point, which is like, and people don't seem to have a problem with this. We mass vaccinate all of the food that you eat. every fucking cow, every fucking chicken. If you eat meat or you consume dairy, you're directly consuming the flesh or the product of animals that have been vaccinated. Straight up.
Immunize wildlife against rabies. We have rabies vaccines for wild animals. There are a whole bunch of them that get rabies, and we put it in bait and drop it from helicopters in the woods, and it drops down the incidence of rabies in people.
Wow.
People hiking get bitten and so forth. It drops the incidence, so we can do that.
I didn't know that. I was wondering how much medical care we're doing for animals in the wild, because I've recently become more and more aware that animals are living in extreme poverty.
Mm-hmm.
Right? Like, you don't know, you think like natural, it's great. Like when animals are living on a farm, it's terrible. But then you also have to compare to like what life is like or like the zoo. You have to compare what life is like in the wild.
Well, life in the wild is very tough, I think. I mean, most animals have to, well, the carnivores anyway, they have to catch their food every day.
Yeah.
Right?
And then there's the viruses there.
They have viruses as well.
Oh,
So
there's that.
the rabies immunization is the only one I'm aware of for wild animals. We do immunize lots of other animals. We immunize chickens and pigs and cows,
Right.
even fish, farmed fish.
Oh, wow, yeah, I didn't think about
you
that.
Pick each fish up and give it an injection, when it's a small fish. But that's mostly so that the farmers get a good yield. We don't really care about the animals, right? We want a good yield for market. And then there's some examples where we immunize animals to prevent spillovers into people. So there's a disease called Hendra in Australia, which
Sounds gnarly.
discovered in the 90s. And it turns out there are bats, fruit bats, that have this virus. And the bats are fine, but sometimes they fly into horse stalls, and the horses get infected. These are, in Australia, it was initially race horses, which are very expensive, right? The horses got infected and they died, and the humans who would take care of them would die also. So now they immunize the horses to prevent to save the horses. Probably that's the motivation, because these horses are hundreds of thousands of dollars, right? And then the people don't get sick because the horses don't get sick. You don't want to immunize all the people because it's too rare, but that approach is called the One World Health approach, which means everything's connected on the planet, and we have to think of everything in the grander scheme, not just us.
Yeah, so you can immunize some things along the trajectory that a virus would take.
Exactly.
So not some living beings. Yeah.
In the Arabian Peninsula, they have a MERS coronavirus issue. Every month there are a couple of cases where a camel will infect a human, and the human can get very sick.
Yeah, there was a really serious outbreak of MERS COVID in like Egypt and Saudi Arabia and countries where like camels are like very prevalently interacting with humans, and it's fucked up, man. It's a horrific. It's a similar respiratory virus. It sucks to get.
It's a respiratory disease, very very much like COVID, and so
sorry, I'm a little bit of a
camels are very
smarty
common
pants.
there. They're raced, they're used as pets, they're eaten. So there's a lot of human camel contact, but the number of cases are rare, two a month. So you don't want to immunize all the humans. So the idea would be to immunize the camels.
Right.
So
I like it. So so you put rabies, but Ebola also is a famously deadly one.
right,
What is it? It kills like, I don't know, 50-60% of its.
could be 50 to 90, but that's
50
in
to
Africa
90.
where the healthcare isn't great. What you saw
I
when
guess that's the
when
point, yeah.
cases of Ebola came to the U.S., we could take care of it. We knew how to take care of it. We had fancy hospitals and so forth, and now we have a vaccine. So we can, and the vaccine is really good, but there are many governments in Africa that are suspicious of us, and they don't want to use our vaccine. So
there's a vaccine for Ebola,
there is, yeah.
and the effectiveness and safety of it, how much is understood.
So this is difficult because there's not a lot of Ebola, right? It's not a continuous ongoing thing. There are sporadic outbreaks here and there.
Of a few thousand people.
At most, usually a few hundred. And the biggest ever, in fact, this is why we didn't for years have an Ebola vaccine. The U.S. military together with Canada, developed an Ebola vaccine for service people, right? They wanted to say, well, we're sending people into these Ebola areas. We want a vaccine for them. So they had developed it through all the preclinical, which means before it goes into people. And that stopped because there was no money to do a phase 1 and a phase 2 and a phase 3. In fact, for a phase 2 and 3, you need to have infections going on because you're looking at how well the vaccine prevents infections, right? So then there was a West African outbreak in 2013, 2015. The most cases ever, 25,000. So they got to test the vaccine. But they only put it in a few thousand people. It's not like it's been in hundreds of thousands of people like the COVID vaccines has been. So it looks like it has high efficacy, but we'd like to have more data. Side effects are not so great. There are a couple of different available vaccines. Some have been tested more than others. I would say this would probably not be widely accepted in the U.S.
But then neither would be something over 50% deadliness of a virus.
No, I think if you were, in fact, many physicians work in countries that have Ebola, so they get vaccinated because they understand the choice.
Yeah,
right.
It's always about the choice. So
then one more thing to answer the interesting, what are some of the viruses you're really fascinated by? There are a number of viruses that have clearly been shown to alter host behavior, and that's how they spread.
Mhm.
I think those are fascinating. For example, there's some viruses of plants that are spread by aphids. And the aphid bites the plant, the virus reproduces in the plant, then it somehow engineers the plant to give off volatile organics to attract more aphids, which will spread the virus. Isn't that amazing?
yeah,
So that's altering the
Reminds me of Twitter.
behavior. Altering because somehow the virus infecting the plant cells gives off these organics
So
and it attracts
interesting.
aphids. And furthermore, somehow when the aphid bites, it tastes horrible. So they immediately leave with the virus they've just picked up and go to another plant to spread it. So they're attracted and then repulsed at the same time.
And obviously, you don't want to anthropomorphize this like a strategy they're taking on. Somehow this worked out.
It worked out this way. It just evolved. And evolution is sometimes hard to trace, right?
Mhm.
Like Darwin famously said, he could never figure out how an eye evolved from a single cell, right? But it did.
The more complicated, complex the holistic organism is that the virus invades, the less able it is to control that organism, right? So I wonder if there's viruses that can control human behavior, to induce more spread of the virus.
Well, I don't see why not.
There's not enough humans, I suppose, to evolve through.
Well, we can't do the experiment to test it, right? We have to observe, and that's always hard when you're observing because there's so many things that can confound what you're looking
Yeah,
at.
change human behavior. Yeah.
I mean, there's so many things that impinge on our behavior, but I think it's possible. I think it's highly possible. If it does it in a plant, why not change some other organism's behavior? I think it's fine. Anyway, those fascinate me. There are lots of examples of those that are fascinating, and how they work, people are trying to figure out. But there's not a lot of money to work on, insect and plant viruses unless you're going to the USDA. So they don't get a lot of work moving forward.
Well, if you understand some of those viruses, is that transferable to human viruses? That understanding?
I think some of it could be, sure. I think the general principles, for example, how does the virus cause volatile organics to be made? It must be turning on some genes, and you could learn principles from that, how the virus might do that. Sure, I think everything is broadly applicable. So to say it's not useful to study viruses of insects and plants is just wrong, because in science, you probably know this, maybe in your field it's the same. If you're curious, you're going to run into interesting things that you never planned on, right?
That's why people you can.
It's kind of the nature of all scientific discovery, right? Is just people getting interested or hung up on a particular thing, or just kind of stabbing in the dark and oh, I think I'm gonna check this out. It's super fascinating.
Criticize why do we want to go on Mars? Why do we want to colonize Mars?
Okay, I think we're going to skip ahead a little bit. I want to hear him talk a little bit about SARS-CoV-2, And then we're going to talk about vaccines.
So we talked about what is a virus. We talked about some of the most dangerous and deadly viruses. Can we zoom in and talk about COVID-19 virus?
Sure.
I don't know what your preferred name is, but
Well,
maybe
there's
for
two
this,
names, right? The virus is SARS-CoV-2,
Yeah.
which is hard, it's long, right? And then COVID-19 is the disease. So you could say the virus of COVID-19, that's fine.
the virus of COVID-19. But for the purpose of this conversation, we'll every once in a while just say COVID.
It's fine. No problem.
What is this virus? From I don't know how many ways we could talk about it. I think from a basic structural, like the virion structure, biological structure perspective. What is it? What are its variants? Can you describe the basics, the important characteristics of the virus?
So virus.
Before we go too deep into, I'm just going to show you guys something cool here. NextStrain is actually a website that tracks all of the global data on the different variants of a variety of pathogens, things like Ebola, avian influenza, seasonal influenza. But obviously there's quite a bit of resources put into SARS-CoV-2. So you can look at, there are different data maps, and if you're a data scientist, there is quite a bit of information in terms of the original genome, where the different sequences happened. You can see delta, alpha, gamma. You can look at, search by specific variant, et cetera, et cetera. So there's an enormous amount of genomic data that you can look into and how it's spread, the number of occurrences, et cetera, et cetera. It's pretty cool stuff.
Viruses are classified by humans just to make it easier to keep track of them, right? So this is a coronavirus, which is because when they were first discovered, I think the first ones were animal coronaviruses. They looked at them under the electron microscope, and it looked like the solar corona, and that's all there is to it. And I have to say,
like a lot of things in science, it kind of has a stupid reason for having the name that it has. If you ever are in a conversation, with someone who is an anti-vaxxer, not even anti-vaxxer, but someone who doesn't believe in COVID, they say, well, COVID's bullshit because if you look on the back of the Lysol wipes that I bought 3 years ago, it says COVID on it. Like it says SARS-CoV. And I'm like, yeah, because SARS-CoV-2, it's a different version of that virus. Like coronaviruses have been around for a long time. They come in different mutations. This is a new one. And those subtleties are sometimes difficult to explain to people and get people to follow, particularly if they don't want to follow it. It makes it infinitely more frustrating.
say that early in the outbreak, the place with the highest seropositivity in the U.S. for a while, 68%, was a working class neighborhood in New York City called Corona. Can you beat that, right?
That's crazy. Yeah.
So coronaviruses, they have membranes, right? We talked about membranes. They have spike proteins in the membrane so they can attach to cells. And inside, they have RNA. And they are the viruses with the longest RNA that we know of.
This is an interesting point, actually.
Close.
The
For some
coronas,
reason,
specifically.
They're able to maintain 30,000. So SARS-CoV-2 RNA is 30,000 bases of RNA. And some of the other coronas are even longer, 40,000.
This is coronas are a family of viruses that included the one you mentioned before, version 1.
So SARS-CoV-1, yeah.
CoV-1, and I guess other ones as well.
So we first learned of them in animals. A lot of animals, pigs and cows and horses have coronaviruses. And then in the 60s, we discovered a couple of human coronaviruses that just cause colds, very mild colds that you wouldn't even think twice about, right? And then suddenly, in 2003, there's this outbreak of severe respiratory disease in China.
This is SARS.
And
this is the mutation of
it
COVID.
It started in November, and they didn't tell the world until February. And that was really bad because it was already spreading by the time they told people about it. But this went to 29 different countries. Only 8,000 people were infected, and then it stopped. And that was the first time we saw an epidemic coronavirus, and what they did afterwards is they said, okay, it looks like it came from the meat markets. They have live meat markets in Guangzhou, in the south of China, where you can go and pick out an animal, and the guy will slaughter it for you and give it to you. And then, of course, there's blood everywhere, and that's how they got infected. And they figured out that there's this animal called a palm civet that was the source of virus. The palm civets are shipped in from the countryside, and the palm civets somehow in the countryside got it from a bat. So they went looking in caves in the countryside, and they found in one cave all the viruses that could make up SARS-1. And that was 2000, and well, I would say took about 8 years after that outbreak. So that was the first hint that bats have coronaviruses that can infect people and cause problems, right? And after that, we should have been ready.
So didn't they already start developing vaccines after then?
So some people started making vaccines. They tested them in mice, but they never got into people. And some people started working on antiviral drugs. Nothing ever came of them because, industry, there's no disease. It's gone. Why should we make vaccines and drugs? And NIH in the U.S., you submit a grant and they say, it's too risky. There's none of this virus around. So people were really short-sighted, because I always say we could have had antivirals for this. Absolutely.
Yeah.
For sure. No question. In fact, the one antiviral that's in phase 3, it's called molnupiravir. It's the only one that you can take orally. It's a pill. It looks really good. That was developed 5 years ago, but never taken into humans. It could have been ready.
So I want to stop here really quickly, because there's an excellent point later in this conversation where they kind of address the whole. There's obviously a huge problem with profit being a motive in healthcare. Period. Like, profit in treating people and looking for treatments for diseases really should be completely removed from the idea of people making enormous sums of money, because when that becomes the motive, obviously the types of investments those companies make shift, right? They don't necessarily have a modality of caring about searching for a wide variety of things, wide variety of treatments, even for incidents of disease that aren't like super high in different populations. And they muse a little bit about this later. I'll let us get to the point, but I'm glad that he addresses it, and that he's like, all we need to do is put money into the right things. And if we point money at these problems, like, a lot of the times we find solutions. It's just about finding the political will or the political motivation to get there.
We dropped the ball, and then the next decade, 2012, MERS coronavirus comes up in the Arabian Peninsula. This comes from camels and infects people, but probably the camels got it from bats originally.
So I want to skip ahead here just in the interest of time, because there's quite a bit of like really good content coming up. We're going to skip right into the part about vaccines. So earlier in the stream, we were looking at how the immune system responds to different pathogens through a variety of different cells that attack and identify and wake up T cells, et cetera, et cetera. We're going to dive into the vaccine portion here, because that's like the meat and potatoes of what I want to get to.
She was asking how difficult is it to make vaccines like that it seems like it's very difficult to do that for the influenza virus.
So it's really easy to make an old school vaccine. So the way the first influenza vaccines were made was actually Jonas Salk worked on them in the 40s. You just grow lots of virus. And you grow it in eggs, by the way, chicken eggs.
Nice. Literally? Wait.
Yeah, embryonated, so they get fertilized, and there's a 10 or 12-day embryo in it, and you put virus in it, it grows up, and then you harvest it. You get about 10 mLs of fluid. And then you take that, you treat it with formaldehyde or formalin, and it inactivates the virus, so it's no longer infectious, and you just inject that into people. And that was the first flu vaccine that was made for the U.S. Army, actually.
Holy shit.
And then it got moved over to people.
Wow.
We still use that old school tech today.
So you're taking. Can you help me out here? Okay, so this is a good time to talk about vaccines.
There we go.
Okay, so you're talking about you're taking the actual virus,
Right.
you put it in an egg, You let it grow up. It's very funny that you put it in an egg. It's very poetic. And then, how do you make it not infection, not effective or whatever?
Not infectious.
Not infectious. Is that the right term here?
Yeah.
So how do you make it not infectious?
You can treat it with any number of chemicals that'll disrupt the particle so it no longer
So
infects.
that
And the point is that if the virus is quote unquote dead and you inject that viral particle into like the bloodstream, your immune system is still able to identify the pieces, the proteins, the little building blocks of that virus, recognize it as like, hey, that's fucked. Like, I'm going to build some T cells that can deal with this if this comes around again. And thus, if you were to experience getting exposed to the live virus, your immune system is going to have the weapons to actually fight that. So like the first rudimentary vaccines were very much this. We're just like injecting people, with like what they thought were dead viruses. And I think at some point he addresses when the polio vaccine first came out, there was a very high occurrence of children that got paralyzed from taking the polio vaccine because they didn't do a good job of killing the virus. There was live virus still in the vaccine, and so when they gave it to a bunch of kids, it just straight up gave them polio. So the process in which you need to do that obviously needs to be rigorously controlled and quality checked.
that step of disrupting the particle, is that very specific to a particular virion particle?
No, the same collection of chemicals you can use for all kinds of,
Gotcha.
which have been used for SARS-CoV-2 vaccines also.
So
Same technology.
Okay, so there's several things to ask. So you called it old school in a
Right.
way that's slightly dismissive, like people talk about
Yeah.
Windows 98 or something. So is there risks involved with it, or is it just difficult to produce large amounts?
No, it's
takes
all
a lot of eggs.
It's very easy. I mean, you could do it in cells and culture, but eggs were convenient, and in the 1940s we didn't have cells and culture. We
Yeah.
didn't know how to do that, so we had to use something else. It's easy to do, but the process of inactivating the virus with a chemical makes it not the best vaccine you can make. The flu vaccines that we have today, which are mostly based on this inactivation, this is called inactivated virus vaccines.
Oh, so like the kind of thing it presents to the immune system to train on is not
great.
it's
It's not
not
great.
it's not com- it's
Yes.
not close to the actual virus.
Yes, that's what we think. So that's why probably the flu vaccines are just not very good. 60% efficiency at the best, right? Which is not really
It's not
good.
very great.
What does it mean? What is the measure of efficiency for a vaccine?
Well, it's how it does in the general population at preventing influenza.
At preventing
Illness, not infection. We usually don't measure infection when we're testing a vaccine. We just measure sickness. That's really easy to score, right?
Mhm.
You
And-
do a trial and you say, if you feel sick, give us a call. We'll tell you
So,
what to do.
yeah, I mean, what's sickness? Sickness is the presence of symptoms.
So this is good time to say what a symptom is. Okay, a symptom is what you only can feel. Only you can feel an upset stomach or a sore throat or that sort of
It's
thing.
the lived experience of
Whereas
a
a
symptom.
sign
Right,
is
right.
something that someone could measure and
Int-
tell that you're infected. Like,
Right. So if they
So
look
obviously they
whoops,
don't know
wow, I
any-
screwed up there. If they actually look inside of your body and like, yo, your shit's inflamed, like you got some issues here, like we can definitely see there's problems versus like, it's like, oh, my stomach hurts, my leg hurts, Sorry, I'm gonna try and find my spot back here. I
Just
screwed
like
up
people
there.
talk about
Yeah.
Windows 98.
process of inactivating, well, it's how it does in the general population at preventing, right?
Mm-hmm.
You do a trial and you say, if you feel sick, give us a call.
Sorry, and one last thing I just want to mention really quickly is this is like predominantly going to be this is one of the big talking points of anti-vaxxers that really drives me like up the fucking wall. Is saying, well, it doesn't even the COVID vaccine, you can still get it. You can still get it. And it's like, yeah, you can. It's not that you still can't contract the polio virus. It's just that the vaccine has a certain level of efficacy in making sure that virus doesn't actually result in signs or symptoms, right? And often that needs to be a lot more of the conversation. The narrative is that the vaccines aren't there to straight up stop COVID in its tracks. It's to keep people out of hospitals. It's to reduce the symptomatic effects. And in some cases, like, some people will straight up just not get sick. Some people will straight up not transmit the virus. But there's a healthy blend of those things that gives us as a society much better protection if we all participate in, this really important. And frankly, it's like, I know people a lot to say, like, oh, this infringes on my freedom and, it's my body, my choice. Well, I don't know. I think that, I'm not much of a patriot, but what I will say is that I think it is civilization. It's our duty sometimes to stand up and do what's right for everybody. And I think, in this particular regard, getting vaccinated definitely falls in that regard.
We'll tell you what to do.
So yeah, I mean, what's sickness? Sickness is the presence of symptoms.
So this is good time to say what a symptom is. Okay, a symptom is what you only can feel. Only you can feel an upset stomach or a sore throat or that sort of
thing. It's the lived experience of
Whereas a sign is something that someone could measure and tell that you're infected, like virus in your nasopharynx or something else, right? Signs and symptoms. And so in a vaccine trial, they tell you if you have any of these symptoms, and they give you a paper with the exact symptoms listed to make sure you're picking them up, right? So for flu, it would probably be fever, sore throat, cough.
Mm-hmm.
You call them, and then they will do a PCR and make sure you've got flu and not some other virus that makes similar symptoms. And then they would say, are you a vaccine or non-vaccine arm? And they count up all the infections and see how the vaccine did, basically.
That's so fascinating because the reporting, so symptom is what you feel.
Yes, for sure.
And certainly the mind has a ability to conjure up feelings.
Oh, yes, absolutely.
And so, like, culturally, maybe there was a time in our culture where it was looked down upon to feel sick or something like that, like toughen up kind of thing.
Yes.
And so then you probably have very few symptoms being reported.
Absolutely.
And then
absolutely.
And now is like much more, I don't know. Perhaps you're much more likely to report symptoms now. It's fascinating because then
self-reporting in medical studies is like it's the huge vast majority of the data that is collected in medical studies comes from self-reporting, from the people who are participating in them, giving up information willingly. And to a certain extent, that's terrifying because you always, think about like, oh, what don't people tell, or what do they forget, or where are they off base on something? So it's interesting to think about.
It changes.
Oh, it is definitely a perception because for, your symptom may be nothing to me or vice versa, right? And so when you're doing this, it's a little bit of a imprecise science because and even it's a cultural thing. In some countries, something that would make us feel horrible, they wouldn't even bother reporting. No, I didn't have any symptoms. So it's a little bit imprecise and it clouds the results.
Okay, we're going to skip ahead here real quick.
Measure things, it's always better.
What are the modern ways? Did you want to look mRNA vaccine? That's
the most modern. But even before mRNA vaccines, we learned that we could use viruses to deliver proteins from a virus that you want to prevent.
Mm-hmm.
And so the Ebola vaccine, we took the spike gene of Ebola virus and put it in a different virus, and we deliver that to people, and that's called a vectored vaccine.
Mm-hmm.
And some of the COVID vaccines are vectors of different kinds. The most famous are adenovirus vectors carrying the spike gene.
I can't remember which one it is. I think it might have been the AstraZeneca one that was adenovirus. It's one of the ones that does not use mRNA. I think Johnson & Johnson maybe as well. And that's one that people would advise, like, if you don't want mRNA, go for that one. It's a more traditional, like, vaccine that, has been used for a significantly longer time in our population, and we have a lot more data to understand its level of efficacy and safety.
Into the cell.
Can you explain how the vector vaccine works again?
So we take a virus that will infect humans,
mm-hmm.
but will not make you sick. In the case of adenovirus, the years and years of people studying it has told us what genes you could cut out and allow the virus to infect the cell but not cause any disease.
So instead of doing selection on it, you actually genetically modify it.
Yes, you modify the vector, yeah.
So you can be much more precise about it.
You're very precise, and then you splice in the gene for the spike, and then you use that to deliver the gene, and it becomes produced as protein, and then you make an immune response.
And vector is the term for this modified.
Right. So we're now using viruses at our bidding. We're using them as vectors, not just for vaccines. We can cure monogenic diseases. That is, if you're born with a genetic disease, you have a deletion or a mutation in a gene, a single gene, we can give.
This is huge, by the way. Like some of the stuff that's coming up in the mRNA space, like its ability to target specific diseases, is extremely promising.
You the regular gene back using a virus vector.
So, but
Cancers too. We can cure cancers with vectors.
wow, really interesting.
Yeah. I think in 10 to 15 years, most cancers will be treatable with viruses. Yeah.
Wow.
And not only can we put things in the vector to kill the tumor, we can target the vector to the tumor specifically in a number of ways. And that makes it less toxic, right? It doesn't infect all your other cells.
But it takes time to develop a vector for a particular thing because it requires a deep understanding.
Yeah, in fact, we have about a dozen different virus vectors that have been studied for 20 years. And those are the set of vaccine vectors that we're using. So it includes adenovirus, vesicular stomatitis virus, which is a cousin of rabies, but doesn't make people sick. Influenza virus is being used as a vector, and even measles virus. So we're familiar with how to modify those to be vectors, and those are being used for COVID vaccines. And then, of course, we have the newest, which is the nucleic acid vaccines. So years ago, people said, why can't we just inject DNA into people? Take the spike and put it in a DNA and inject it. So people tried many, many different vaccines. And in fact, there are no human licensed vaccines that are DNA vaccines, although there is a West Nile vaccine for horses that's a DNA-based vaccine.
Crazy.
So if you have a horse, you can give it this vaccine, but no human.
Can you clarify, does a DNA vaccine only work for DNA viruses?
No, it can work for DNA or RNA, because remember, for an RNA virus, we can make a DNA copy of it.
Right.
And it will still, when you put that DNA in a cell, it goes into the nucleus.
Okay, right. So you're
and
just skipping
eventually
a step
you get proteins
for RNA
made.
vaccines.
Yeah.
You're
Okay,
giving.
interesting.
Okay, I got it.
So those didn't work for human vaccines. And there were many HIV/AIDS vaccine trials that used DNA vaccines. Didn't work. And then, a number of years ago, people started thinking, how about RNA vaccines? And when I first heard this, I thought, what? I've worked with RNA my whole career. It's so fragile. If you look at it the wrong way, it breaks.
It's
I
interesting.
mean, that's being facetious, right? But you have to be very careful because your hands are full of enzymes that will degrade RNA. So I thought, how could this possibly work, injecting it into someone's? It's an example of I was skeptical and I was wrong. It turns out that if you modify the RNA properly and protect it in a lipid capsule, it actually works as a vaccine. And people were working on this years before COVID came around. They were doing experimental mRNA vaccines, and there were a couple of companies that were working on it. And so at the beginning of 2020, they said, let's try it. And
Perfect time to pause here for another video that I wanted to go through, which talks a little bit about mRNA transcription and how mRNA works. And I think it's important to just cover this very quickly. It's only a 10-minute video. It'll help us kind of visualize a little bit better about what is going on when you get injected with an mRNA vaccine. What is actually happening in terms of the biological processes. So we're checking out Khan Academy, great resource if you guys are interested in learning more about science, math, pretty much anything. I can't recommend it enough. So we're going to dive in real quick just to start. Here we go.
Video is a little bit of a deep dive on transcription. And just as a bit of a review, we touch on it on the video on replication, transcription, and translation. Transcription in everyday language just means to rewrite something or to rewrite some information in another form. And that's essentially what's happening here. Transcription is when we take the information encoded in a gene in DNA and encode essentially that same information in mRNA. So transcription, we are going from DNA to messenger RNA.
This is a biological process that is your entire everything uses to build the proteins that are required for day-to-day life.
And we're going to, in this video, focus on genes that code for proteins. So this first step is the transcription, the DNA to messenger RNA. And then in a future video, we'll dig a little bit deeper into translation. We will translate that information into an actual protein.
So in the case of the vaccine, the protein that is being synthesized is the spike protein, which is the specific thing that connects to the ACE2 receptor of your cells and actually allows it to get a foothold so that it can then puncture into your cell and infect it.
These diagrams give a little bit of an overview of it. It's a little bit simpler in bacteria. You have the DNA just floating around in the cytosol. And so the transcription takes place. You start with that DNA, that protein coding gene in the DNA, and then from that you code the messenger RNA. You see that in that purple color right over here. And then that messenger RNA can be involved with the ribosome, and that's the translation process to actually produce the polypeptide, to produce the protein. In eukaryotic cells, and we're going to get into a little bit more depth in this video, the transcription, the DNA to mRNA, that happens inside of the nucleus. And there's essentially two steps here. You go from DNA to what we would call pre-mRNA. Let me write that down. Pre-mRNA, which is depicted right over there. And then it needs to be processed to turn into what we would call mRNA, which then can leave the nucleus to be translated into a protein. So now that we have that overview, let's dig a little bit deeper into this and understand the different actors and understand, if we're talking about a eukaryotic cell, what type of processing might actually go on. So right over here, we are going to start with the protein coding gene inside of the DNA, right over here. And we are going to, the primary actor that's not the DNA or the mRNA here is going to be RNA polymerase. It's used to create a sequence that will become a nucleotide sequence that will become the messenger RNA. So this RNA polymerase, it needs to know where to start. And the way it knows where to start is it attaches to a sequence of the DNA known as a promoter. And every gene is going to have a promoter associated with it.
So the idea is the promoter is essentially hey, here is where the code that you need to copy is, because that's what your DNA is. It is a large instruction set for all of the little pieces that your body needs to build for your body to be your body. And in those sequences, it tells the DNA says, okay, this sequence for building this protein starts here, ends here. And the cells have a way of identifying that, stripping it away into mRNA, which then goes into your ribosomes, and that is what converts that messenger RNA into a protein. And this is the entire process that encapsulate how the messenger RNA vaccine works. So I just wanted to do a very, very quick visualization of that so we can dive a little bit deeper into the mRNA vaccines that are
I was skeptical,
for
frankly,
COVID.
because I just thought RNA would be too labile, but I was wrong.
So this is, as we were saying offline, one of the great things about you is you're able to say when you were wrong about intuitions you've had in the past, which is a beautiful thing for a scientist. But, I still think it's very surprising that something like that works, right?
I am surprised.
So you're just launching RNA in a protective membrane.
Yeah.
And then now one thing is surprising that the RNA sort of lasts long enough, right?
That's right.
In its structure. But then the other thing is, why does it work that that's a good training ground for the immune system. Is that obvious?
Well,
That should work?
I don't think it's obvious to most people, and it's worth going into because it's really interesting. I mean, first of all, they wrap the RNA in fats, in lipid membranes, right? And the particular formulation they test for years to make sure it's stable. You know, it lasts a long time after it's injected. And the two companies that make the current COVID vaccines, right, Moderna and Pfizer, they have different lipid formulations to get to the same. So that's a real part of it. And it's not simple. There are quite a few different lipids that they put into this coating. And they test to see how long they protect the RNA after it's injected, say, into a mouse. How long does it last? And the way it works is these lipid nanoparticles, they get injected into your muscle, they bump into cells, and they get taken up. So fat is sticky. It's greasy, we like to say. And so your cells are covered with a greasy membrane also. So when these lipid nanoparticles bump into them, they stick and they eventually get taken up. And they figured this out right at the beginning. If we put RNA in a lipid nanoparticle, will it get taken up into a cell? And the answer was yes. It was just, let's try it, and it worked.
So it's basically experiment. It's not like some deep understanding of biology. It's experimentally speaking, it seems to work.
Yeah. Well, they had some idea that lipids would target this to a cell membrane. And remember, there's no receptor involved. Like the virus has a specific protein that attaches to a receptor.
Right.
It's not efficient enough to just bump around and get into a cell. That's what these things are doing, and they probably optimize the lipids to get more efficient uptake. But it's not as efficient as a virus would be to get into a cell.
So you have no specific. I mean, which is why it's surprising that you can crack into the safe with a hammer or with some fat. I mean, that's kind of surprising. It's kind of amazing that it works. But so maybe let's try to talk about this. So one of the hesitancies around vaccines, or basically around any new technology, is the fact that mRNA is a new idea, and it's an idea that was shrouded in some skepticism, as you said, by the scientific community, because it's like it's a cool new technology. Surprising that it works. What's your intuition? I think one nice way to approach this is try to play devil's advocate and say both sides. One side is why your intuition says that it's safe for humans, and what arguments can you see if you could steelman an argument why it's unsafe for humans, or not unsafe for humans, but the hesitancy to take an mRNA vaccine is justified.
So many people are afraid because it's new technology and they feel it hasn't been tested. I mean, in theory, what could go wrong? This is the nice thing about mRNA is that it doesn't last forever, as opposed to DNA, which doesn't last forever, but it can last a lot longer, and it could even go into your DNA, right?
There can be a
mRNA
crossover.
has a shorter lifetime, maybe days after it's injected into your arm, then it's gone. So that's a good thing because it's not going to be around forever. So that would say, okay, so it's sticking around for your lifetime is not happening. But what else could happen? Well, let's see, the protein that's made, could that be an issue? And again, proteins don't last forever. They have a finite longevity in the body. And this one also lasts perhaps at the best a few weeks.
Now, this is a protein that's made after the RNA gets into the cell.
Yeah, so the lipid nanoparticles taken up into a cell and the mRNA is translated and you get protein made.
And there's also a question, and I'm sorry to interrupt,
Yeah,
like where in the body, so because it's not well targeted, or I don't know if it's supposed to be targeted, but it can go throughout the body. That's one of the
Right, so
concerns.
it's injected deep into your deltoid muscle, right, your shoulder. And the idea is not to put it in a blood vessel, otherwise it would then for sure circulate everywhere. So they go deep in a blood vessel, and it's locally injected. And they did, before this even went into people, they did experiments in mice where they gave them 1,000 times higher concentrations than they would ever give to people. And then when you do that, it can go everywhere. Basically, you can find these nanoparticles in every tissue of the mouse. But that's at 1,000-fold higher concentration, right? So I think at the levels that we're using in people, most of it's staying in the muscle, but sure, small amounts go elsewhere.
Could there be a lot of harm caused if it goes elsewhere? In like, let's say ridiculously high quantities. I'm trying to understand what is the damage that could be done from an RNA just floating about.
So the RNA itself is not going to be a problem. It's the protein that is
The protein.
encoded in it, right?
So-
This is a viral RNA which has no sequence in us, so there's nothing that it could do. It's the protein that I would say you could ask, what is that going to do? And the one property we know about the spike is that it can cause fusion,
This is a very
of
good point
cells.
he makes here.
right? That's how the virus gets in the beginning.
Right.
The spike attaches to the cell by this ACE2 receptor, and it causes the virus and the cell to fuse. And that's how the RNA gets out of the particle.
But so wait, I'm a bit confused. So with this mRNA vaccine, with the lipids and the RNA, there's no spike, right?
The mRNA codes for the spike,
Oh, the mRNA codes,
Yes.
so it creates the spike,
creates a spike.
and so that spike could cause fusion of cells.
Yes,
potentially.
except they modified the spike so it wouldn't.
Got
They
it.
made 2 amino acid changes in the spike so it would not fuse.
So they understand enough which amino acids are responsible for the fusion.
That's right.
Interesting. This is so.
So
cool.
they could modify
Okay.
it. So now it's not going to cause fusion, so that's not an issue. It's called the pre-fusion stabilized spike.
Cool.
So the spike, when it binds ACE2, that top falls off, and the spike, and the part of the spike that causes fusion is now exposed, and that doesn't happen in this mRNA vaccine. So those are the things that could have happened, but I think they're ruled out by what we've just said. But there's no better test than putting it into people, right?
Right.
And doing phase 1, phase 2, and phase 3 in increasing numbers of people and asking, what do we see? Do we have any concerns? And so now it's been in many millions of people, and we don't see most of the effects you see in a vaccine, you see in the first couple of months. Things like the myocarditis with some of the vaccines, the clotting issues with the AstraZeneca vaccine, Guillain-Barré, you see those relative.
So this is a really important point that I wanted to touch on, and I'm glad that this kind of like precipitated when they mentioned this. When we have conversations with our peers and our friends and our families, and if we're talking about things like vaccine safety, I think it's really important to separate the conversation into 2 very specific categories. The first category is short-term safety. Is the vaccine safe from a short-term perspective? The answer to that question is an unequivocal yes. We have hundreds of millions of people now who have been injected with these vaccines. If it was like changing our DNA and turning us into lizard people, we would see that by now. So that brings us to the framing part number 2, which is long-term, are the vaccines safe? And there's people like Brett Weinstein who will say, there's just no way to know. There's no way to know, so we can't say yes. And that is definitely a standard that we can follow. We can say, yeah, we don't really know, so I guess we can't say yes, they're safe long term. But we don't have any evidence to think that right now, and it's something that we still need to continue to watch for. So I don't think it is a good excuse to say, well, I'm not going to get it because we don't know the long-term effects. Like, the chances of long-term effect are fairly low given the type of technology that it is. And again, it outweighs the risk of dying of COVID, right? Which is something that you could definitely do.
Relatively quickly. And we've seen small numbers of those occur, but other things we haven't seen, and you never say never, right?
Right. So
Right.
I mean, this is fascinating, right? It's like I drink I put Splenda in my coffee, and has supposedly no calories, but it tastes really good. And despite what like rumors and blogs and so on, I have not seen good medical evidence that it's harmful to you. But it's like it tastes too good.
We're going to take a quick pause here to switch gears to another video because we're talking about vaccines, vaccine safety. So let's talk about the side effects of vaccines. Another Kirksall video, super fun, 10 minutes long. And then I think that'll be a good opportunity for us to wrap the stream tonight, unless people want to talk about something more specifically. But chat's been pretty quiet. That's fair. If you're a lurker, if you're just hanging, respect the hell out of that, guys.
Vaccines are celebrated for their part in fighting disease. But a growing group of people seem to believe that they endanger our health instead of protecting it.
Also, I think this video came out pre-COVID. So this was like, yeah.
The internet is full of stories about allergic reactions, the onset of disabilities, and even death following vaccination appointments. And it's true, vaccines can have side effects. Let's look at how they work and how dangerous they really are. Your immune system is a complex army of billions of cells, soldiers, intelligence cells, and weapons factories. Every day you're attacked countless times, but your immune soldiers alone usually deal with that, so you don't even notice. If an infection becomes serious though, our intelligence cells gather intel about the attackers and activate our weapons factories. You know the weapon: antibodies. They're like targeted missiles, produced specifically to combat the invader. Unfortunately, this process takes several days to complete. That gives intruders a lot of time to do damage. Contrary to popular wisdom, what doesn't kill you doesn't make you stronger. Our bodies really don't want to fight serious wars over and over, so our immune system came up with an ingenious way to get stronger and stronger over time. If we fight an enemy that is dangerous enough to trigger our heavy weapons, our immune system automatically creates memory cells. Memory cells remain in our body for years in a deep sleep. They do nothing but remember. When an enemy attacks for a second time, the slumbering memory cells awaken and order coordinated attacks and the production of antibodies. This is so fast and effective that many infections you beat once will never make you sick again. You might even be immune against them forever, which is also why little kids are constantly ill. They don't have enough memory cells yet. And this beautiful natural mechanism is what we build on when we use vaccines. How vaccines work. As great as memory cells are, obtaining them through an infection is unpleasant and sometimes dangerous. Vaccines are a way of tricking our bodies into making memory cells and becoming immune to a disease. They pretend to be a dangerous infection. One way of doing this is to inject invaders that can't do harm. For example, by killing them or by ripping them into pieces. Our immune systems deal with these kinds of vaccines pretty easily. Sometimes it's necessary to make our immune system work harder though to produce even more memory cells. Live vaccines are the real deal. An enemy that can punch back is a bigger challenge than a dead one. But this also sounds like a sort of horrible idea. What if the germs win? To avoid that, we breed a sort of weak cousin of the real germ in the lab. Just powerful enough to annoy the immune system and create enough memory cells. Okay, so these are the basic principles of vaccine use. They provoke a natural reaction in our bodies that makes us become immune against very dangerous diseases. Some, like the flu virus, mutate so often that we need a new vaccine every year, but most vaccines protect us for years, or even a lifetime. But there's a catch. Like everything in life, vaccines have another side: side effects. What are they, and what happens if your child develops one? The risks of vaccines. It's complicated to directly compare the side effects of vaccines with the effects of diseases. For example, hundreds of millions of people are vaccinated against measles in the West, but there were only 83,000 cases in Europe in 2018. So with numbers that different, even mild side effects can seem scary compared to the bad effects of a disease that we don't see as much anymore. Before the measles vaccine became available in 1963, virtually every single child on Earth contracted measles at some point.
Jesus.
An estimated 135 million cases in the 1950s, every single year. But are measles really that dangerous in the year 2019 with our advanced healthcare and new technologies? Are they worth the risk of vaccine side effects? Let's do a thought experiment based on real numbers. Imagine a developed country in a parallel world. It has good healthcare, but people stop vaccinating. In this scenario, let's say 10 million children caught measles. What happens? 9,800,000 or 98% will get a high fever and a very unpleasant rash. Up to 800,000 of them, or 8%, will suffer from dangerous diarrhea. 700,000 or 7% will suffer from an ear infection, which can lead to permanent hearing loss. 600,000 kids, or 6%, will suffer from pneumonia, the most dangerous effect of measles. It alone will kill 12,000 children. Up to 10,000 children, or 0.1%, will get encephalitis. 2,500 kids, or 0.025%, will contract SSPE, a disease where the measles virus lingers in their brain and kills them a few years later. Taken together, around 2.5 million children will suffer from somewhat serious effects from measles, and about 20,000 children will be killed by measles. It doesn't stop here, though. The kids who beat measles are left with a severely damaged immune system that needs a lot of time to regenerate. Time for other diseases to do further damage. Aside from that, what's almost guaranteed is that your kids will have a really bad time for about 2 weeks. Okay, but what about vaccines? It's only fair to look at these risks too. So let's repeat our thought experiment, but this time we vaccinate 10 million children with MMR. What would theoretically happen here? After being vaccinated, of our 10 million kids, around 10% get a fever. 500,000 or 5% will get a mild rash. Up to 100 or 0.001% might have a serious allergic reaction and will have to be treated. Up to 10 boys or 0.0001% might get inflammation of the genitalia.
Okay.
And up to 10 kids in total, or 0.0001%, might get the most serious side effect, encephalitis. So we vaccinated 10 million kids. In total, around 120 had side effects that were somewhat serious. Thanks to good medical treatment in developed regions, almost all of them will be fine. What about autism? The connection of autism and vaccines stems from one source that has been disproven countless times. We'll link to further reading and videos in the description. But in 2019, it's fair to say that vaccines do not cause autism. All right, but what about deaths? It's really hard to say if even a single one of our 10 million vaccinated kids would die. We really did look hard and talk to a lot of different experts. If we ignore self-reporting, we could only come up with a tiny handful of documented cases for all the hundreds of millions of children who received the MMR vaccine since 1971.
Wow, there you go. Self-report.
Measles are many, many thousands of times more dangerous for your child than even the absolute worst case of vaccine side effects. You have to search for lethal vaccine side effects with a huge magnifying glass, and even then, it's very hard to find confirmed cases, while deaths from measles are real and easy to find. In 2017 alone, 110,000 people died from measles worldwide. Statistically, today 300 children will die of measles. One since this video started. You can compare vaccines to seatbelts. Are there weird freak accidents where someone gets killed by their seatbelt? Well, yes. But do you personally think it's safer to not put a seatbelt on your kid? Wait a second.
Probably
What
not.
if your kid is actually allergic? What if none of the things we've said applies to your specific situation? In this case, you need to become the greatest vaccination promoter of all. Because if your children can't be vaccinated, only the collective can protect them. This is called herd immunity,
There we go.
and it's the only thing that can protect your unvaccinated child. Herd immunity means that enough people are immune to a disease that it can't spread and dies before it reaches its victims. But to accomplish this for measles alone, 95% of the people around you need to be vaccinated. Conclusion. The problem with the debate about vaccines is that it's not fought on a level playing field. While the pro-vaccine side argues with studies and statistics, the arguments against them are usually a wild mixture of gut feeling, anecdotes and misinformation. And feelings are often immune to facts. We'll not convince anybody by screaming at them, but we can't hide
very
from the
good
reality of
point.
what anti-vaccine conspiracies do. They kill babies too young to be vaccinated. They kill healthy children that are just unlucky. They bring serious diseases back from the verge of extinction. And the biggest side effect of vaccines is fewer dead children. Vaccines are one of the most powerful tools we have to eradicate the monsters that most of us have already forgotten. Let's not bring the beasts back.
It's so fucked that they did this like months before COVID. All right, guys. I think that's a good place to take a break for tonight. Been at it for a couple of hours. We're going to keep picking up the conversation on mRNA, vaccine, safety. We're going to keep watching the next conversation. Until then, guys, thank you for joining. I really appreciate having you all around. And keep looking at the stars. Take it easy.
Oh, boy, oh, boy.
Good company.
Well, pull-out alert. Hi, Mark. Big pull-out alert. Left alert.
Wait, let's try to shoot this guy.
Shoot this guy. That's true.
Got the helo. I had a friend drop with me. I don't see where he went.
Alright, let's get Mark in the game.
Where is he? Where is he?
Oh, I killed him.
Okay, nice.
Chase him down.
Alright. Ball header fun now.
Go.
Matt, you pulling us out?